Hello students, today we shall do the second part of lesson 10, cell and cell organelles. In the previous video, we have learned the components of cell, that is the cell wall, plasma membrane and cytoplasm. We have also seen two cell organelles, that is the nucleus and endoplasmic reticulum. Today we will be learning few more cell organelles, that is the Golgi complex, lysosomes, mitochondria, vacuoles, plastids, chloroplasts in detail. Golgi complex. It is made up of 5 to 8 hollow and flat sacs placed parallel to each other. These sacs are called cisternae and are filled with different enzymes. The proteins coming from endoplasmic reticulum are enclosed in vesicles which come towards Golgi complex via cytoplasm. They fuse with the formation phase of the Golgi membrane and empty their contents in the cisternae. As they pass through the cisternae, they are chemically modified with the help of enzymes. They are again packed in the vesicles. These vesicles exit from the maturation phase. Thus, cisternae works like a packaging department that packs and distributes substances. Now let us look at the functions of Golgi complex. Golgi complex is the secretory organ of the cell. It modifies, sorts and packs materials synthesized in the cell, that is the materials like enzymes, mucus, proteins, pigments, etc. and then dispatches them to various targets like plasma membrane, lysosomes, etc. It produces vacuoles and secretory vesicles. It helps in the formation of cell wall, plasma membrane and lysosomes. Now we will see the next cell organelle, that is the lysosome. Due to various metabolic activities in the cell, organic waste is generated and it is the work of this lysosome to digest the waste. Lysosomes are simple, single membrane bound sacs filled with digestive enzymes. Now let us look at the functions of lysosome. First one is immune system, that is, it destroys viruses and bacteria that attack the cell. Second, demolition squad, that is, it destroys all the worn out cellular organelles and organic debris. It is also called as autolysis. Third, suicide bags, that is, when a cell becomes old or is damaged, lysosomes burst and the enzymes digest their own cells. And the last one is, during starvation, lysosomes digest stored proteins and fats. Each cell requires energy and this energy is produced in the mitochondria. Under the electron microscope, a mitochondria is seen as a double membrane structure. The outer membrane is porous and the inner membrane is deeply folded. These folds are called cristae. The inner cavity is filled with proteinaceous gel like matrix containing ribosomes, phosphate granules and DNA. Therefore, it can produce its own proteins. With the help of enzymes, mitochondria oxidize carbohydrates and fats in the cell. The energy released in this process is stored in the mitochondria in the form of ATP, that is adenosine triphosphate. Plant cells have less mitochondria than animal cells. Now let us look at the functions of mitochondria. First, to produce energy-rich compounds ATP, that is the adenosine triphosphate. The other function is synthesis of proteins carbohydrates, lipids, etc. by using the energy in ATP. Here you can see the ATP molecule. The next cell organelle is the vacuole. Vacuoles are the storage sacs for solid or liquid contents. They don't have any typical shape or size. The structure of vacuole changes according to the need of the cell. Vacuole is bound by a single membrane. Functions of vacuole. First, to maintain the osmotic pressure of the cell. To store metabolic byproducts and end products, that is the glycogen, proteins, water, etc. In animal cells, they store water, waste products and food, while in amoeba, it stores food before digestion. In plant cells, vacuoles are full of cell sap and provide turgidity and rigidity to them. When the vacuole is full of the cell sap, you can see it gives a rigid structure to the plant. An empty vacuole causes the cell wall to shrink, and that results in the droopy structure of the plant. Have you ever wondered why are plants' leaves green and the flowers red, yellow, orange or blue? The organelle which gives such colors is present only in the plant cell, and it is called the plastid. Plastids have double membrane and they are of two types. That is the chromoplast or the colored plastids and leucoplast, that is the white or colorless plastid. Chloroplast can get converted into other types of chromoplast. For example, raw green tomatoes turn red due to the breakdown of chlorophyll and the synthesis of lycopene. The raw green color of tomatoes are due to the pigment called chlorophyll, and they turn red. It is because of the pigment called lycopene. Now let us learn about some pigments and the color which they impart to the plant part. The pigment chlorophyll gives green color to the plant, and that is why we see the leaves are green in color. The pigment carotene gives orange color to the plant. For example, carrot. It is because of carotene the carrot has orange color. Xanthophyll gives yellow color to the plant, and that is why we find yellow color flowers or fruits with yellow color. Anthocyane gives purple or blue color to the plant. The pigment betanin gives dark pink color, for example beetroot. And the pigment lycopene gives red color. For example, we see the red color in tomatoes. Chloroplasts. Chloroplasts are important for the photosynthesis process that takes place in the leaf. The pigment chlorophyll in chloroplast traps solar energy and converts it to chemical energy. You can see stroma in the diagram. Stroma in the chloroplast contains enzymes, DNA, ribosomes and carbohydrates that are necessary for photosynthesis. Functions of chloroplast. Chloroplast converts solar energy to chemical energy during the process of photosynthesis to provide food to the plant. Chromoplast gives different colors to flowers and fruits. Leucoplasts are involved in the synthesis and storage of food like starch, oils and proteins. The organelles mitochondria and plastids can produce proteins as well as replicate themselves, as they have their own DNA and ribosomes. After studying the cell and cell organelles, you must have come to know that all the functions in plants and animal cells are taking place smoothly due to the cell organelles. developed cells are called eukaryotic cells. Last year, we have studied prokaryotic cells of bacteria. Now let us compare them. Eukaryotic cells are of the size 5 to 100 micrometer. They have more than one chromosomes in them. Their nucleus has a nuclear membrane, nucleolus and nucleoplasm. Mitochondria and plastids are present in them. And these eukaryotic cells are present in highly evolved unicellular and multicellular plants and animals. If you look at the prokaryotic cell, their size is very small, that is 1 to 10 micrometer. They have only one chromosome and nucleus is almost absent. You can just see a nucleolide which resembles the nucleus. They don't have a membrane-bound cell organelle. And the example of prokaryotic cell is the bacteria.